Hey guys! Today we're going to be talking about organic molecules, specifically nucleic acids. So first of all, what are nucleic acids? They are large molecules that are like information molecules. They carry genetic information within the sequence of the smaller units that make them up. So what they do is they encode and transmit genetic information. That is their main function. And it's important to note that nucleic acids are polymers. That means that they are large molecules that are made up of smaller repeating units. And they are polymers of nucleic acids. Those are the smaller units that make them up. And the two main nucleic acids that are found in our cells are ribonucleic acids and deoxyribonucleic acids. So the first one is RNA. Here is a diagram of what it would look like, and we can fill it in. So the whole thing is the ribonucleotide. And there are three units that make it up. So the first one, this is the ribose sugar. So this whole thing is the ribose sugar. And you can see by the name it's ribonucleic acid. And this one has an oxygen atom, which is connected to a carbon, which is connected to one carbon and another carbon on the other side, and those carbons are connected to another carbon as well. So with this, there is one oxygen and four carbons, and they are numbered. So the first carbon is called 1' carbon, the second one is the 2' carbon, 3rd' carbon, 4th prime, and then here is the 5' carbon, which is a CH2. And the CH2 is attached to an oxygen, which is part of the phosphate group, and that is this dotted part right here. This is what we call the phosphate group. And on the other side, right here, the carbon is connected to a base. And the base for would be either A, T, C, or U. That's the base for the ribonucleic acids. And the last important thing to note is that all of the carbons are attached to hydrogens or other carbons, and the main difference between the ribonucleic acid and the deoxyribonucleic acid is that the 2' carbon is attached to an OH, and it's different in the ribonucleic acid, and we'll get to that. So, on the other hand, when we have the deoxyribonucleic acid, it's all pretty much the same. There's an oxygen connected to two carbons, and then those carbons are connected to hydrogens or carbons as well. And the same thing applies with numbering the carbons, 1', 2', 3', 4', and 5' carbons. And the main difference is that the 2' carbon is not attached to an OH. It is attached to only H. And you can already see that based on the name that it makes sense, because this is ribonucleic acid, and here it's deoxyribonucleic acid. So there is one less oxygen. That's why it's OH here and just an H here. And the same thing applies with this dotted section being the phosphate group. So this is the phosphate group. And there's a base here as well. And there is a difference between the bases, because for the ribonucleic acids, it's either A, T, C, or U. But here, it's either A, T, C, or G. And this whole thing here is called the deoxyribo, no, it's not writing, nucleotide. Okay, now that we've discussed the structure of the ribonucleotides and the deoxyribonucleotides, we can talk about their bases. So the bases are built from nitrogen-containing rings, and there are two types. There's the pyrimidine bases and the purine bases. So first, with the pyrimidine bases, they have a single ring, like all three of these, and there's three different kinds. So the first one is the cytosine. This is the C. And then the second one is the thymine. And this is a T. And the last one is the uracil. And this is the U. And it's important to know which, whether they're found in DNA or RNA. The cytosine and the thymine, both of them are found in DNA and in RNA. But the uracil is only found in RNA. It's only found in RNA sequences. And now we can talk about the purine bases. So the purine bases have two rings. It's a double ring structure. And there's two kinds here. So the first one is guanine. And this is the G. And the second one is the adenine. And this is the A. And for both of these ones, they are both found in DNA and in RNA. So both of them are found in both sequences. And now that we've discussed that, we can talk about the phosphodiester bond and nucleotide sequences and how they make a double helix. Okay, so now we can look at what these nucleotides would make. So it's important to note that the sequence of the nucleotides determines the kind of information that is stored in the DNA or in the RNA. And with a phosphodiester bond, we are able to connect the adjacent pairs of nucleotides when the phosphate group in one nucleotide here covalently bonds with the sugar in another nucleotide. That'd be this one. In the first nucleotide, we have this phosphate group, and then the sugar, and then the base. That's one nucleotide. And then we have another nucleotide here, where there's the phosphate group here, and the deoxyribose sugar. And there's a covalent bond between the phosphate group and the sugar of both of these nucleotides, and they make a phosphodiester bond. So when we have a long sequence of these phosphodiester bonds between nucleotides, we're able to create a double helix. So this is when there's two strands of nucleotides around each other, and it's in the form of a double helix, and that's what's commonly found in DNA. And here we have a diagram of that. So there's these two strands, and we can look at it in terms of this. So the black long strand, if we just look at one of them, would be this exactly. And the other strand would be the same thing, except if we have this strand and then another one of that, but they're facing each other like this, and then like this actually. And then my fingers would represent the base pairs, which adjacently join. So here we have the colored sections are the complementary base pairs, shown here as AT and CG, and it makes the double helix, as we said. And the black part is the sugar phosphate backbone, which makes perfect sense because this is the sugar phosphate backbone. It's a phosphate and the sugar, and it just continues with the phosphodiester bond. And this is just in DNA because the AT and GC are the complementary base pairs. But if it were in RNA, then it would be GC pairing together, and instead of AT, it would be A to U and G to C. All of this would be in RNA. This is DNA. And that's pretty much the video. So thanks for watching, guys. Today we learned about organic molecules, nucleic acids, and the simpler units that make them up, and how they make DNA and RNA. Stay tuned for more. Thanks for watching, guys.